The human body contains around 20,000 proteins, each with specific functions that keep all of us alive and healthy. One of these particular proteins is insulin. Insulin is a hormone, which means it is released into the blood flow as a signal to different body parts that it is time to perform a function. Insulin is released from the pancreas into the blood flow to tell muscle, fat, and liver cells to absorb a sugar called glucose so it can be used as energy or stored as fat. People with diabetes, however, do not have adequate insulin to keep these blood sugar levels safe. That is why they have to give themselves insulin. People with type 1 diabetes need fast-acting insulin, known as prandial insulin, whenever they eat, and people with type 2 diabetes need slow-acting insulin called basal insulin throughout the day. Today, we're going to take a look at a type of basal insulin called N-Lithocholyl insulin. N-Lithocholyl insulin is very similar to normal insulin. It has an A chain and a B chain, and it is capable of forming normal dimers and hexamers. However, what makes this strain of insulin special is, you guessed it, lithocholyl. What is lithocholyl? What does it do? Let me back up for a second. Proteins are made up of amino acids, which are essentially little building blocks. On the N-Lithocholyl insulin, the threonine amino acid at the end of the B chain was removed. This leaves a lysine amino acid at the end of the B chain. Because threonine was removed, the lysine has a spare nitrogen atom that is not bonded to anything. Here's where things get exciting. Lithocholyl, a nonpolar fatty acid, bonds with that nitrogen. So now we have an almost normal insulin molecule with this fun little fatty acid attached to it. That's cool, but what's the significance? Great question. There are a few important parts here. First, as I mentioned earlier, this is a basal insulin analog. This means it slows insulin activity. The lithocholyl acts as a second reception point, only it bonds with serum albumin, a protein in blood plasma, instead of insulin receptors. Because now there are two reception points, only about half of insulin molecules can do their job at a time because the other half will be bound to these serum albumin proteins. The second thing that makes N-Lithocholyl insulin so special is that it won't crystallize. In hexamer formation, or when 6 insulin molecules come together, most insulin analogs crystallize or form concentrated clumps of hexamers. This doesn't happen with N-Lithocholyl insulin. The nonpolar lithocholyl fatty acids surround the insulin, making the whole hexamer effectively nonpolar and therefore soluble in water. The soluble exterior prevents hexamers from crystallizing together and dissolves any crystals that happen to form. Crystalline insulin is very inconsistent in its rate of dissolving, so if people take their insulin before they go to bed, they really don't know how long it will take to set in, so they could have complications due to irregular blood glucose levels. This sets N-Lithocholyl above other basal insulin analogs and makes it a very viable tool in the treatment of diabetes. Thanks for watching.